Good day, students. Now we will look at the role of the terms given in cell division. First of all, we will look at the chromatin. Chromatin is a complex of DNA and protein that forms chromosome within the nucleus of eukaryotic cell. Nuclear DNA does not appear in free linear strands. It is highly condensed and wrapped around nuclear proteins in order to fit inside the nucleus. Under the microscope, in its extended form, chromatin looks like beads on a string. The beads are called nucleosomes. As you can see in the picture here, each nucleosome is composed of DNA wrapped around 8 proteins called histones. So quickly we go and see the examples of application of chromatin. During interphase 1, chromatin in its least condensed state and appears loosely distributed throughout the nucleus. Chromatin condensation begins during prophase 2 and chromosomes becomes visible. Next, chromatin. Well, what is chromatin? During DNA division, when a cell divides, it needs to take its DNA and duplicate it and then transfer half of it to one cell and another half to other cell. Well, DNA is arranged in chromosome as you know, so what's happened is, as a chromosome replicates or makes a copy of itself, it arranged as two chromosomes next to each other called chromatid. Here you can see it is arranged next to each other. So application of chromatid. Chromatid allows cells to store two copies of their information in preparation for cell division. This is important to ensure that daughter cells are healthy and fully functional. Next, chromosome. Chromosomes are thread-like structures located inside the nucleus of an animal and plant cell. Each chromosome is made up of protein and a single molecule of deoxyribonucleic acid. It is passed from parents to offspring. DNA contains the specific instructions that make each type of living creature unique. So examples of application during mitosis, the chromosomes which have already duplicated condense and attach to spindle fibers that pull one copy of each chromosome to opposite sides of the cell. The result is two genetically identical daughter nuclei is formed. Next, sister chromatids. Sister chromatids are two identical copies of the same chromosome formed by DNA replication attached to each other by a structure called the centromere. During cell division, they are separated from each other and each daughter cell receives one copy of the chromosome. Next would be the homologous chromosome. Sister chromatids are two identical copies of the same chromosome formed by DNA replication attached to each other by a structure called centromere. It's like the centromere is the attachment center for the sister chromatid. During cell division, they are separated from each other and each daughter cell receives one copy of chromosome. Next would be tetrad chromosome. Each pair of chromosome called a tetrad or a bivalent consists of 4 chromatid. Okay, first we know sister chromatid. Here you can see it's like it has only one chromatid. Then at this point the homologous chromosome exchange genetic material by the process of crossing over. When the process of crossing over occurs, the chiasma here Okay, next we will go and see the tetrad chromosome. Each pair of chromosome called a tetrad or bivalent consists of 4 chromatids. At this point, the homologous chromosome exchange genetic material by the process of crossing over. Next would be the centromere. So, the centromere is the specialized DNA sequence of a chromosome that links a pair of sister chromatids. During mitosis, spindle fiber attached to the centromere via the kinetochore. So finally we will see the kinetochore microtubule. Kinetochore microtubule attached to the chromosomes to the spindle pole. So the interpolar microtubule extends from the spindle pole across the equator almost to the opposite spindle pole, and astral microtubule extends from the spindle pole to the cell membrane.
There are two types of cells in a sexually reproducing organisms. First is the gamete cells. Second is the somatic cells. To define the gamete cell, first we need to know how we form. Gamete cells are similar to reproductive cells. Sperm, which is a haploid cell, fused with egg, ovum, also a haploid cell. This fusion is called fertilization and form zygote. Sperm and ovum are the gamete cells, and this type of cells produce through meiosis. Another type of cells is the somatic cells. All the cells in an organism except the gamete cell are the somatic cell. Somatic cells are produced through mitosis. For example, muscle cells, nerve cells, stomach cells, and etc. You also can refer to the images given. These cells are inherited from each parent, which means after the formation of the zygote divide mitotically and share same characters with their parents. Our genetic information is carried by the chromosome, which is located in the nucleus. It determines whether it is a haploid or diploid cell. Each somatic cell in human body consists of 46 chromosomes or 23 pairs and considered diploid. Diploid cells present in two sets of chromosomes, one from each parent. Diploid cells are also denoted as 2N. They are the result of the mitosis and identical with their parent cell. Haploid. Haploid are the cells which consist only half of the chromosomal number. They are appeared singly and denoted as N. They are not identical with their parent cell and result of meiosis process. For example, if the number of chromosomes in the diploid cell of the cat is 38, then the number of chromosomes in haploid cell of the cat is 19. This can also written as N equal to 19. Crossing over is the exchange of genetic material that occurs during cell division. Crossing over is also a very important process in life because it helps to provide variation among organisms. Before the crossing over, in earlier stage of meiosis 1, two homologous chromosomes come together to cross over. This process is called synapsis. Then only the homologous chromosome undergoes crossing over, which is the exchange of the genetic materials between the non-sister chromatids. Chiasmata is the point at which the segments of non-sister chromatids cross over. Eventually, this point will be visible due to the exchange of genetic material after the homologous chromosome apart from each other. The absence of chiasmata leads to improper chromosomal segregation. When we compare karyokinesis and cytokinesis, karyokinesis is the division of the nucleus during cell division, while cytokinesis is the division of the cytoplasm during cell division. Besides that, karyokinesis is the process will divide the nucleus into two genetically equal and identical daughter nuclei. On the other hand, cell division process complete on cytokinesis is done. Karyokinesis is independent to cytokinesis, while cytokinesis is dependent to karyokinesis. Microtubules are originated from centrioles. There are three types of microtubule: astral microtubule, polar microtubule, and kinetochore microtubule. In the beginning of cell division, all these microtubule appears as spindle microtubule. This spindle microtubule provides a framework and means of attachment that keeps chromosomes organized, aligned, and assorted during the entire process of mitosis.